Bioindicators are species that can forecast environmental quality based on their presence, absence, abundance, or physiological status. They offer assessments of ecological conditions and detect changes before traditional methods. Bioindicators include plants, animals, microorganisms, and biological reactions, providing insights into environmental effects. Markert, Brewer, and Zechmeister, 2003, defined bioindicators as organisms, or parts of organisms or communities, that contain information on environmental quality. They provide data by studying the effects of environmental changes on organisms or analyzing the chemical composition of the organisms, reflecting environmental conditions. This definition emphasizes the importance of bioindicators in indicating ecosystem health and wider environmental changes. Epiphytic bryophytes are non-vascular plants that grow on other plants, primarily trees, without deriving nutrients from their host such as mosses, liverworts, and hornworts. Related parameters for epiphytic bryophytes include 1. Air quality, concentration of atmospheric pollutants such as SO2, NOx, and heavy metals. 2. Humidity and microclimate conditions. 3. Forest canopy structure and light availability. Characteristics that make epiphytic bryophytes suitable as bioindicators are: bryophytes are highly sensitive to airborne pollutants such as sulfur dioxide, nitrogen oxides, and heavy metals due to their method of absorbing water and nutrients directly from the air. These plants depend on high humidity and stable microclimates, making them good indicators of microclimatic changes. Bryophytes lack a vascular system, so they have a direct relationship with atmospheric conditions and can quickly reflect changes in air quality. The presence, abundance, and diversity of bryophytes can reflect the health of the forest canopy and overall ecosystem integrity. For Case Study 1, monitoring air pollution in urban areas using bryophytes. Urban areas often suffer air pollution from vehicle emissions, industrial activities, and other sources. Monitoring air quality is crucial. Epiphytic bryophytes are used as bioindicators to assess urban air quality due to their sensitivity to pollutants. Researchers studied urban and peri-urban areas, collecting epiphytic bryophyte samples from tree bark. They measured air pollutants and analyzed the health of bryophyte species in relation to the air quality. The research found that in areas with high levels of pollution, there was a decrease in bryophyte diversity and abundance due to sulfur dioxide and nitrogen oxides. This led to the absence of sensitive species. In contrast, areas with lower pollution levels exhibited higher bryophyte species diversity, indicating better air quality. As a conclusion, epiphytic bryophytes are effective indicators of urban air quality. They are sensitive to airborne pollutants and easy to sample, making them valuable for monitoring and mitigating air pollution. In Case Study 2, assessing forest canopy health in Borneo using bryophytes. Borneo's rainforests are rich in diverse plant and animal species, but are threatened by logging, deforestation, and climate change. Monitoring their well-being is crucial for conservation efforts. Epiphytic bryophytes found in the canopies can indicate forest health and microclimatic conditions. Researchers conducted surveys in primary and secondary forests in Borneo, collecting bryophyte samples from different canopy levels. They assessed the diversity and abundance of bryophytes in relation to canopy structure, light availability, and humidity. Microclimatic conditions, including temperature and humidity, were also measured. The study showed that primary forests with intact canopies have more bryophyte diversity compared to secondary forests. Species like Frelania and Radula were particularly abundant in primary forests, indicating stable microclimatic conditions and high humidity levels. In contrast, secondary forests with fragmented canopies showed reduced bryophyte diversity and more desiccation-tolerant species, reflecting altered microclimatic conditions. Epiphytic bryophytes were effective indicators of forest canopy health in Borneo. Their sensitivity to microclimatic conditions and dependence on stable humidity levels make them valuable for monitoring the impacts of deforestation and forest degradation. Epiphytic bryophytes are chosen as bioindicators due to their sensitivity to air pollution, humidity, and forest canopy conditions. They reflect environmental quality through their presence and diversity. Case studies in urban areas and rainforests show that bryophytes are effective bioindicators, providing valuable insights for environmental monitoring and conservation efforts.